Okay, this lecture is about mitosis. Basically, mitosis is how we divide chromosomes to create two identical cells, which we call daughter cells. So here are your objectives. We'll go through those again at the end of the lecture. Let's remind you about chromosomes. So we are going to be focusing on mitosis, meiosis, and genetics of humans. So humans have 46 chromosomes in 23 pairs, and pairs 1 through 22 are called your autosomes, which are just your regular old chromosomes. And then the 23rd pair are your sex chromosomes. And sex chromosomes determine your biological anatomy. Right? Whether you'll have male parts, female parts. Humans' sex is determined generally as XX is female, XY is male. When you get into your upper division genetics course, you'll learn that there are always exceptions to the rules. And this image of your chromosomes is called your karyotype. The chromosomes are numbered in pairs from the largest to the smallest, and then the sex chromosomes are always put together, even though they don't really pair up quite the same. They're of different sizes, as you can see. Humans are considered diploid, or 2N. That means we have 2 of every chromosome. So our chromosomes are in sets of 2. So you can see we have 2 number 1s, 2 number 2s, 2 number 3s. That's because you get 1 from your mom and 1 from your dad. So you have a maternal mom and paternal dad chromosome for every pair. Even your sex chromosomes, you would get 1 from your mom and 1 from your dad. So if you're XX female, you get an X from your mom and an X from your dad. And if you're XY, you get your X from your mom and your Y from your dad. And we will see this more in genetics. So diploid means we have 2 sets of chromosomes. Haploid is talking about the number N, which would mean half of the chromosomes in a body cell. So haploid for humans are only in gametes, which is your reproductive cells, like your egg or sperm. So the diploid number for humans is 46. 2n equals 46. So haploid is 23. So you just take 46 divided by 2. If you have any kind of deviation from this 46 chromosomes, you're called aneuploid. So aneuploid means you have a different number of chromosomes. So in this case, this person has 3 chromosomes, number 21. So they are considered aneuploidy, which just means they've got either extra or less chromosomes than the typical human. You are going to see diploid numbers a lot. So if you see 2n, say, equals 4, you know that that cell has 4 chromosomes in 2 pairs. So you just take this number, divide by 2, and that gives you your pairs. So a 2n equals 16. You have 16 chromosomes, 8 pairs, right? So you'd go 1, 2, 3, 4, 5, 6, 7, 8. 8 pairs. Okay. Not all organisms are diploid. Some could be triploid. Tri means 3, so you have 3 sets of every chromosome. This is not viable in humans, but a lot of plants have what we call many, like polymer, polyploidy. In fact, strawberries, most of the strawberries you eat are octoploidy. That means they have 8 sets of chromosomes. So they actually have 7 different chromosomes, just like you have 23 different chromosomes, but each chromosome is represented 8 times. So they'd be 1, 2, 3, 4, 5, 6, 7, 8. There's 1. 1, 2, 3, 4, 5, 6, 7, 8. One, 2, 3, 4, 5, 6. Oh, we need to give him one more. I think that's right. Anyways, lots of chromosomes. Plants can tolerate this. Most mammals cannot. Okay, so make sure you understand ploidy. Diploid is 2. Triploid, if I gave you something, this would be 8n equals 56. So you would know that one set of chromosomes has 7 different chromosomes. So we'll work with those numbers. Really important terms you've got to keep straight. Homologous chromosomes versus your sister chromatids. So, homologous chromosomes is a pair of chromosomes. So these are, I'm giving you examples of 3 homologous chromosomes, 3 pairs. Okay. Homo means the same in biology. So they have the same type of information. So for instance, this might be the one from your mom and this is the one from your dad. And they both might have information about eye color, and they both might have information about, say, skin color. Where on this homologous pair, they might have information about your height. So what you inherit from your mom and dad is not identical. But each chromosome pair has the same type of information. So they have the same genes, but can have different alleles, versions of those genes. Okay, so homologous chromosomes are not identical. They have the same type of information, but because you're inheriting them from different people, they're not the same. Now, after a cell undergoes S phase in cell cycle, which is DNA replication, then your chromosomes look like X's. So it's a duplicated chromosome held together at the centromere. And we'll talk about centromeres in just a minute. So we now say that these two are sister chromatids. And they are identical because they're due to DNA replication. So these are two identical copies. And so let's say it's this one. And then your dad also got duplicated. And so you have two identical copies. So these are also sister chromatids. But these two X's with the same amount of information are still homologous. Okay, so we just took them and we replicated them. They're still considered homologous chromosomes because they have the same type of information. They're not identical, but the sisters that were copied are identical. So, whenever you see, let me write this in a different color. Whenever you see this X, that's standing for two sisters. So right after replication, they're held together by this centromere region. Okay, so cell cycle we have talked about. I want to emphasize to you that from G1, S, where your DNA is replicated, to G2, this whole part is called interphase. It says it right here. There's another phase of cell cycle, and that goes from here to here, and that's called M phase. And M phase includes mitosis, which is what we're going to talk about, and cytokinesis. So mitosis is separate from cytokinesis. Mitosis is the division of chromosomes into two cells. Cytokinesis is the division of the cytoplasm into two cells. Okay. So they both make up M phase. Don't let this fool you. It's not just mitosis. M phase is mitosis and cytokinesis. Mitosis, we're going to learn, has 4 phases called PMAT. So, watch these terms. Make sure you know the differences. Alright, so let's talk about the super cool thing called mitosis. And the next lecture is going to be on meiosis. So there are some web pages about the differences and similarities between mitosis and meiosis. Super important, could be a nice compare-contrast question. I've linked lots of different videos for you to actually watch mitosis in action. We're going to draw it out, but it's pretty cool to watch. So let's talk about the purpose of mitosis. The purpose is to make 2 identical cells. So for humans, we are going to start with 1 diploid cell. And I want you to think for a minute, what is the diploid number for this cell? And I hope you figured out 2n equals 2. You just count the number of chromosomes. So this only has 1 pair, 1 from mom, 1 from dad. After DNA replication, which is S phase, hopefully I didn't start that. You've got your sister chromatids as little X's, right? Still same number of chromosomes, 1 homologous pair. Then we're going to talk about mitosis, how we divide, and the end result is 2 diploid cells. So you started with 1 diploid cell, you doubled the DNA, and then you split it into 2. Okay. The purpose is to make identical cells. Let's make sure we understand about our mitotic chromosomes, because you're going to see these little X figures a lot. Down here, again, it shows you this is a pair of sister chromatids. One chromatid, one chromatid. They are identical because they have just been duplicated. And some terms you need to know. The first one is a centromere. The centromere is an area or region of the chromosome where the sisters are joined together. Okay. So this is important. It's an area or region. What actually is joining them is the kinetochore proteins. Okay, so these help keep the sisters together. And this is where spindle fibers, which we'll talk about in a minute, attach. So the kinetochore is a series of proteins that sits at that centromere region, allows the sister chromatids to keep together until cell division is ready, and it's where the spindle fibers attach. So let's look at what spindle fibers are. Okay. So here's another term for you. The centrosome, again, is an area or region where the spindle fibers form. And they form from the organelle called the centriole. So when we drew it, we drew it like a pair of tubes. So these were microtubules, part of the cytoskeleton. Okay, you can kind of see they're in pairs here, and they generate these long microtubule fibers called spindle fibers. So there's lots of different types. There's astral, there's polar, there's kinetochore. You don't need to worry about the different types. You just need to know that spindle fibers help pull chromatids apart. So they connect to that kinetochore and during cell division, you're going to see movement. They're going to pull those sisters apart to opposite ends of the cell. All right, so you know about the chromosomes, you know about some of these technical terms. Let's look at mitosis. Okay. Oops, I forgot to move this. So we're going to come back to this. Let's look at it first, and then we'll come back and draw it. Interesting. So I guess I didn't save the one I fixed. Okay. We'll keep going. All right, so first, before we start mitosis, here is interphase. I actually had made these larger so you could read them, but oh well. Okay, so interphase, remember, is G1, S, G2. And what I want you to see in these images is the blue are chromosomes. And what I want you to see about interphase is you cannot distinguish the chromosomes. It just looks like a blue mass. And it's important to understand this because this is going to help you identify with images. So, interphase, after S phase, the chromosomes have replicated. They're all this tangly mass. What happens next is what I call, or what is called, prophase. Now, some textbooks talk about pro-metaphase. Honestly, you cannot tell the difference. We are not cytologists. We're not trained in this. So we're just going to combine these two. And so prophase, what I want you to be able to recognize is that you can see chromosomes. So you see this is, let me erase this. This is blobby blue, right? Now you can start to see those squiggles. Because the chromosomes have condensed, which means they've gotten tighter. Okay. The nuclear envelope is going to break down. So it says here, nuclear envelope. Breaks down. And spindle fibers form. So in these images, the green is representing, or is the staining of, the centrosome. So you can kind of see this area, and the green is also representing those spindle fibers. So they're starting to form, they're starting to connect to the chromosomes or the sister chromatids, right? And they're going to do a little tug of war until everything lines up in the middle, and that is then called metaphase. So remember, meta stands for middle. So in metaphase, the chromosomes are lined up in the middle. So you can kind of see, if you look at the difference between here they're kind of spread out and here they're all condensed in the middle. That's metaphase. If you remember back to our cell cycle lecture, there is a, I know stop signs have 8, right? There is a stop or pause in the cell cycle. There's a checkpoint. That's the word I was looking for. And the cell pauses here in metaphase to make sure that there is spindle fibers attached to every chromosome, so that when we go to separate, we can separate those sister chromatids. We're going to draw it like this with the spindle fibers to make it a little easier to see. Okay. Next phase is anaphase. So we've talked about prophase, which is the P. Metaphase, anaphase, telophase is the M-A. Telophase, T. Okay. So anaphase is just when you start to split the sisters. Okay, so you can see now they're all here, they're lined up nice in the middle, and now they're starting to separate. So you get separation. Telophase is the last phase of mitosis. And so in this phase, the chromosomes decondense, so they get more spread out. And you can kind of see it's harder to see nice long squiggle lines like you can in anaphase. And the nuclear membrane begins to reform. So you can see these dotted lines. Okay, that's really hard to see under the microscope, but what you want to see the difference is that you can't really clearly see the chromosomes anymore. They're like a blob, and they're starting to separate. The final part is cytokinesis. So remember, this is not part of mitosis. This is the splitting of the cytoplasm. And it's slightly different in animal versus plant cells because we don't have cell walls. So in animal cells, you can kind of see that these cells, they start to split. The plasma membrane is growing, and we have this what's called a cleavage furrow. And eventually these cells split into two. It's different for plant cells because they're not going to totally split in two. Instead, they make a cell plate that is going to become the cell wall. And so eventually you're going to have a nice cell wall here that separates those two cells. Okay? Cytokinesis completes the cell cycle, and we have now gone from one cell to two. So, what I want to do with you is I want to, draw this. So, mitosis, we're going to draw for a cell that's 2n equals 4. And what you're going to see is in class and quiz and tests, I'm going to give you a 2n number. You're going to figure out the chromosomes. Now, the easiest way I think is to draw in all different colors. So, I'm going to say 2n equals 4. Right now, all we have is a bunch of squiggles. Right? You can't tell the difference in interphase. Even though we have, all right, let's see if I can do this. I'm going to make a red. Oops. Oh, I forgot I was going to make a red, a blue, this is going to be a pain, a green, and an orange chromosome. Okay, 2n equals 4, 4 chromosomes. And then what we have to do is undergo S phase and duplicate all of these. Right? So I'm going to have my duplicated orange, my duplicated red, my duplicated blue, and my duplicated green. And we don't really see these until prophase. So let's put them in here. We've got them all squiggling around. And what color do I need? Oops, I forgot my orange. Alright, so prophase, you can now see the squiggles, whereas here it was all just mushed together. If I drew every color, right? Okay, metaphase, line up in the middle. So what I want you to do is I want you to line them up. So you've got your red chromosome, you've got your blue chromosome, you've got your green chromosome, and you have your orange chromosome. You can line them up horizontally or vertically because this cell is splitting vertically. I think that's how it is. This is why I'm lining them up this way. Okay, let's also draw our spindle fibers. So here is our centrosome, that area that these spindle fibers have come from, and there's one on this pole or opposite end of the cell as well. And so what's going to happen in anaphase is that the spindle fibers are going to get shorter and they're going to pull the chromosomes to the opposite end of the cells. So our red is going to be here. Do you see how I just split that sister? And my blue is going to be here. And my green and my orange, right? So we split the sister chromatids. In telophase, we're going to complete that splitting, and we're going to start drawing that cell membrane. So now I have one red chromosome at each end of the cell, one blue chromosome, one green, and one orange. Here is your PMAT. Prophase, metaphase, anaphase, telophase. This is mitosis. Cytokinesis completes the cell cycle. So now we're going to have our nuclei formed. We're going to actually split these cells, and they each have, oops, can't change the color, one of each chromosome, just like the original cell. So here was the original cell, right? We replicated the chromosomes and then we split them. And so now we have two identical cells. Just looks just like what we started with. And yes, they'll start to decondense and look like squiggles. Color them however you want, squiggle, 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 and they'll look like a mush. Okay, but I drew them because I wanted you to understand that we've made two identical cells. That's mitosis. You need to practice understanding the 2n number that I give you, and you need to practice drawing these phases. So on an exam, I will ask you, I will say a cell has 2n equals blah number, blah, and draw me, say, anaphase, or telophase, or metaphase, prophase, okay? I won't ask you to draw the whole thing, I'll ask you to draw one phase, and that shows me, do you know the number of chromosomes, and do you know what's happening at that phase? The other thing you'll have to do is be able to recognize images of mitosis. So let's try that. Oops. Okay. So again, you can pause this. I want you to name the phase of mitosis shown here. And hopefully you can convince yourself this is metaphase. So in fact, those chromosomes are lined up in the middle of the cell. All of these cells that you can't distinguish, like say this one, this is interphase. They just look like they've got dots. Those dots are some condensed or I shouldn't say condensed. They're just regions of the nucleus that's staining dark because there's a lot of chromosomes there. But you can't see the squiggles. So if you can't see the squiggles, that's interphase. Okay, try this one. Okay, so this cell right here is showing anaphase. I want you to be able to see that the chromosomes are separating. Okay. This one would also be anaphase. This one I wouldn't give you because it's hard to tell, but this is probably metaphase. This one I would say is probably prophase. And then, for instance, these cells are an interphase. These cells, if you can see they're smaller, they just divided. So this is a plant cell. The classic way to look under the microscope is at the growing root tip of an onion. You know, the green onions you can buy in the store. If you leave them in your fridge a long time, they'll grow these root tips out of the white part, which is the part that you usually eat, along with the green, I guess, but at that end. And if you had a microscope at home and you had some stain, you could see mitosis happening. Probably don't, so you'll have to just look at pictures on the internet. All right, how about this one? If the original cell has 22 chromosomes, how many chromosomes do each of the new daughter cells have? The answer is 22, because the daughter cells are identical to what you started with. So if you start with 22, you're going to make 2 new cells with 22. So you're going to go to 22, you're going to double that to 44 chromatids, and then you're going to split that back to 22 and 22 chromosomes. So you'll see numbers. Not heavy math, but you got to figure it out. All right. Oh, well, don't click A because we're not doing that. But can you put these in order? So the answer's on the next slide. So if you haven't done it yet, pause. So the answer goes from interphase, where you cannot distinguish chromosomes, to prophase, where you can start to see the squiggles, to metaphase, where it's lined up in the middle, anaphase, telophase, where they're really apart and starting to get smushy. And then I know that this is the last one because you see how these cells are smaller than this one? So that's cytokinesis. So again, lots of activities and resources for you to look at. Let's just finish this off with a little bit of why do cells divide? Why do cells do mitosis? Okay. So I have posted a BioFlix film on mitosis. I encourage you to watch it. It's a really nice animation. And some of the reasons is we do this to grow. So you were once a little kid, now you're a big kid, and you had to grow. And the way you grew was not by making bigger cells, because we already know we need to keep that cell surface, large cell surface to volume, small volume ratio. So you had to make more cells. Repair. So if you damage your body, you can repair it by making more cells through mitosis, and sometimes just replacement. So for instance, your blood cells are replaced every 1 to 5 days. That's why you can donate blood quite often. Your skin, depending on where it is, every 10 to 30 days. That's why you shed all this skin, dandruff if you have it up on your scalp, right? And remember, you're leaving your skin cells behind, so don't commit any, do any bad things, because your DNA is in there. Taste buds, every 10 days you get new taste buds. So don't worry, when you drink that hot chocolate and burn your tongue, those taste buds will grow back. Your small intestines, because they work so hard, they get damaged every 2 to 4 days. You've got new small intestine layer. Pretty amazing. So all of the cells in your body are dividing by mitosis. Once you became a fertilized egg with sperm, a little zygote, you underwent mitosis, 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 mitosis. Okay. This is called asexual reproduction. So mitosis is an example Because you're making identical cells. So this is bacteria. Bacteria don't divide by mitosis. Technically, it's called binary fission because it's a little more simple than mitosis. But what you're doing is you're taking one cell and making two identical. So it's like cloning. You're making identical copies. Many protozoa, like my friend Euglena or Paramecium or amoeba, will do asexual reproduction where they'll just grow bigger and split in half. And now we've got two. This is compared to sexual reproduction, where this is a combination of genetic information from two sources. And what you're making is something genetically unique. The way you do sexual reproduction is you make you make cells that have N chromosomes, haploid, and they combine to make a 2N zygote. So this process of making these haploid cells is called meiosis, and that is the next video that you will watch. All right, thanks.